All right, here are your notes on viruses' structure and function. The first thing you need to know is that viruses are the smallest infectious particle. They're absolutely tiny, and we'll talk more about that in a second. And viruses aren't technically alive by definition, because all the things that are required for you to be alive, they possess, except they can't reproduce on their own. So because they can't reproduce on their own, they are not technically alive. The typical size of a virus is roughly 100 nanometers in diameter. And here's a look at what a 3D rendering of a virus. They don't actually look like this in real life. We can't take that clear of a picture that small, but that's roughly what a virus would look like. To give you an idea of what 100 nanometers in diameter is like, an erythrocyte, which is a red blood cell, is roughly 8 micrometers. Nano is a billionth, micro is a millionth. So to give you an idea, if this was 10 micrometers in diameter, then a red blood cell would be 100 times larger than a virus. So viruses, very small. Obviously, I can say these things faster than you can write them, so it's possible that you may need Jess to pause the video. So please feel free to do that at any point in time. All right, next up we have virus structure. The structure of viruses looks like this. There's a capsid. Every virus has a capsid, and there is our friendly virus again. And every virus has a capsid, and that capsid is what stores the genetic material. Viruses may have an envelope and save room. See, this gap is here because I'm going to draw a picture here. So make sure that you save some room to draw a picture. So viruses may have an envelope, and an envelope has virulence factors on the outside of it. You can think about those in terms of antigen and antibodies, and they work best when they fit together. And so it's how the virus identifies itself, and it's actually how the virus attacks our cells. So the virulence factors look like this. They're like tiny little antigens, little protein markers on the outside of the shell of a virus. Then inside that envelope, and remember the envelope might be here, might not be here, but what definitely is there is the capsid. The capsid is the little protein container that has all of the genetic material inside of it. If you haven't paused already, I would give people a chance to pause and write this down, because you don't want to try and listen and draw at the same time. You'll miss what you're listening to. All right. So then the genetic material is housed inside the capsid, and there is our little strand of double-stranded RNA. And the thing about viruses is that they are different than people. People must have double-stranded DNA, that typical alpha helix that we've all seen many times now. There it is. That is double-stranded DNA. Just this one right here, double-stranded DNA. But viruses can have single-stranded RNA, double-stranded RNA, or single-stranded DNA. They can use any type of genetic material they want. Oftentimes, viruses that use RNA as their genetic material are referred to as retroviruses. HIV is a good example of a retrovirus. All right, so capsids come in different shapes as well. Capsids are always present, and a virus may or may not have that membrane around it, or that envelope. And the shape of capsids vary, and we'll talk about that in a moment. Here's an example of how viruses use envelopes. So the virus envelope attack. Here is a virus with a little envelope around it. So the red is the envelope, the black hexagon is the plasmid, or the capsid, and inside is the single-stranded RNA. So the way this works is that the proteins on the outside of this envelope bond to a very specific protein on a human cell. So here's an intact human cell sitting around minding its own business. The virus will move toward it, recognize the proteins on the outside of that cell, and bond to it. So here is the envelope of the virus that has fused itself to the envelope of the human cell and created an opening. And inside that opening, it dumps its capsid and its, inside the capsid, in this case, single-stranded RNA. And it is going to move over here, insert itself into our genetic material, and it's going to start using our ability to create new DNA against us. So where the RNA primase and RNA polymerase comes in and creates a new RNA chain from our DNA template strand, it's going to insert itself right in there, and our body will actually start making copies of the virus for it, so that the virus can then multiply inside of our cell, eventually burst our cell, and leave and go infect other cells. And there you go. The envelope attaches, injects the capsid with DNA or RNA. In this case, I used a single-stranded RNA virus. Remember that you can pause at any point in time. Maybe at the end of notes is the best time to pause so that people can catch up on the drawing. All right, then we're going to talk about types of virus. You name viruses for a host of reasons. You can name them based on their nucleic acid, their shape, their structure, or the method in which they replicate. We're going to talk about 3 of the main viral types now. The first one is a bacteriophage. The bacteriophage you might remember from the Hershey-Chase experiment. This little lunar lander setup does exactly that. This whole molecule will land on the surface of a cell with these little feet down. And then a tiny little needle-like structure drops out of there and punctures into our cell, and it injects its DNA, or in this case, double-stranded RNA. RNA is red, just for these notes, not in general. And it injects it into the cell. So that's a bacteriophage. The next kind is a picorna, and a picorna is just a capsid, no membrane, but it's an interesting capsid in that it actually opens up once it gets inside of a cell, and that's how it releases its, in this case, single-stranded RNA. And the last one is the herpes virus, and the herpes virus attacks with an envelope, and it has all of these antigens out front so that it can find a particular cell, bond to it, fuse with its membrane, and open up, just like we saw here. So it would use its envelope to bond to the envelope of a cell and inject it. That's the herpes virus. All right, last but certainly not least, there are some viruses that infect specific cells based on the presence of suitable receptors. So that's what we're talking about with the envelope and its antigens on the outside. HIV is a classic example of that. HIV has all of these little triangularly shaped appendages, virulence factors, on the outside of its envelope. When it comes in contact with a helper T cell, its triangularly shaped virulence factors match perfectly with a cell receptor on the outside of the T helper cell. At that point in time, it bonds to the membrane, opens up the membrane when it fuses with it, and injects the virus into the helper T cell. All viruses need another living cell in order to replicate. It's why viruses are technically not alive, because they lack the ability to reproduce on their own. Those are notes. Watch them as many times as you need. Email me if you have questions. I'll be back next Tuesday. I miss you guys. See you in a few days.